my voice?
Yes, ma'am. Good morning.
Okay. On the last class, we studied about action potential. So it's the transfer of impulse along the neurons, which could able to conduct the stimulus. So here, I also displayed some videos about the action potential, but in YouTube you can search some more videos and you can search some more important points. Okay, I think it's available in both languages, Tamil and Singalam. So these all things you already completed on your A levels, but just you're again studying these things. So you can get more knowledge if you access to the YouTube videos and if you learn some more, that will be much beneficial. After learning from the YouTube, if you connect it with the notes, you can understand better. Okay. So here today we are going to study about under the heading of functions. So here we are going to again repeat some more notes about the action potentials. Okay. So just I'm asking some questions. If you know, you can answer. And what is the resting membrane potential value? Okay, very good. Okay. And what is the threshold value? So after this threshold value, the depolarization occurs.
Minus 55.
Okay, very good. And why the depolarization happening? Okay, opening of which canal causing the depolarization? Okay, fine. I think it is better to do this practicals physically. Okay, on online, you may, I think you're not listening much, and this is not a good way to teach the subject. So that's why I put some videos. Anyway, I don't know about other subjects, but about the action potential, I will repeat on the physical class. Okay. If you are having physical class, I will repeat this portion, action potential. Okay. All the others are just note that you can read and understand, but here you have to explain it through some graphs. Anyway, I will repeat it. Okay, now we can study about the functions. So again, when the neuron is not conducting any impulses, there is some voltage variation. There are some charge variations in between the voltages. Okay, that is the resting membrane potential. That is about minus 70 millivolts. So here on the first point, nearly all the cells from animals, plants, and fungi will function like batteries. Batteries means, in the batteries you can see the positive and negative volts. So, in the sense that they are maintain voltage difference between the interior and exterior of the cells. Okay, when the cell is at resting membrane potential, sodium ions will be higher outside than the inside, and potassium ions will be high inside than the outside. So because of this, there is some voltage difference between the interior and exterior of the channels. Okay. But interior side, they will have more negative poles or negative charges. When the voltage of a cell is measured in millivolts or thousands of a volt, so typically the voltage of the animal cell will be minus 70 millivolt, the resting membrane potential. As the cells is so small, the small magnitude voltage give rise to strong electric forces within the cell membrane. Okay, Because the cells are so small, the voltage of this magnitude give rise to very strong electric forces within a cell membrane. Okay. About this slide, it is also just repeating what we learned earlier. Okay. Just in this slide, you must remember that there is voltage difference between interior and exterior of the cells. When the neuron is not conducting the impulses, it is about minus 70 millivolts. Okay, that's enough about this slide. In the majority of cells, the voltage changes vary very little over time. So the change of voltage is very small in majority of the cells, but there are some sense of their voltage fluctuations. Okay, in some type of cells, they will be electrically active when the voltage fluctuations means the changes occurs. Some of these voltages sometimes show a rapid up and down fluctuations that have a stereotype form, up and down cycles that are known as action potentials. Okay, all these slides are explaining. So the previous slide and this slide is explaining what we learned earlier. Okay, and just you must know that interior part and exterior part of the cells have voltage difference. At resting membrane potential, the voltage is about minus 70 millivolts. And in some cells, the voltage fluctuations occurs, and the up and down cycles of these fluctuations are known as action potentials. The duration of this action potential vary across a wide range. Okay, the duration can vary when considering the brain cells. Okay, the cells that are available in the brain, the entire up and down cycles will be less than a thousands of seconds. Okay, so the very minute, the entire up and down cycle duration. But in some other type of cells, this will last several seconds. Okay, again I'm explaining. The duration of action potential can vary cells to cell. Okay, in brain cells, the action potential duration is very short. Okay, thousands of a second. Okay, that means a fraction of a second. But in other type of cells, this action potential takes several seconds. Okay, so here you must remember the duration of action potential can vary. The electrical properties of an animal cells are determined by the structure of the membrane. Okay? The electrical properties of animal cells are determined by structure. Structure means enabling of lipid molecules. Okay, phospholipids with large protein molecules in between them. So this structure of membrane is deciding the electrical properties of the cells. Okay, I will again explain what is in the slide. Please listen. It is about the duration of action potential. Okay, in some cells the action potential duration may be very short. In some cells the action potential duration may take some long time. The electrical properties of the cell is depending on the cell membrane structure. Okay, normally the cell membrane is made up of a layer of lipid molecules with large protein molecules embedded on it. So the electrical okay, I'll ask the questions. Anyone? Which part of the cell is responsible for the change in electrical properties of cell? Can anyone answer this question? Which part of the cell is responsible for the variations or change in electrical properties?
Cell membrane.
Brain of the cell.
Thank you so much. At least one person is answering. So this membrane is made up of lipid molecules and large proteins within those molecules. Okay. Here the lipid layers of the cell membrane is highly resistant to the movement of electrically charged ions. So they will not allow the electrically charged ion to pass through them. They are resistant to the movement and they are insulating. But in between the cell membrane, there is largely impeded, means in between there's some other molecules are fixed in between the membranes that are protein molecules that are having some channels. Channels means pathways. Through the pathways, ion can pass. Okay, these ions can pass. And some of the large molecules are capable of actively moving some type of ions from one side of the membrane to the other. Okay. So here you must know the electrical properties of the animal is depending on the structure of the membrane. When considering the structure, the membrane is made up of lipid layers, but in between the lipid layers, you can find the protein molecules. And these lipid layers will not allow the ions to pass through them. But the embedded protein molecules have some channels through which the electrically charged ion can pass. Okay? The large membrane embedded molecules, in contrast, provide channels and pathways through which ions can pass across the membrane. And some of the large molecules, there are some large molecules that are capable of actively moving specific types of ion from one side of the membrane to other. So these ions can move through the channels, and there are some other molecules which can actively transfer these ions. Okay. And processes in a typical neurons. All cells of the animal body tissues are electrically polarized. That means they are maintaining a voltage across the cell's plasma, and that is known as the membrane potential. So the animal cells are having membrane potentials across their cell plasma membranes. And this electrical polarization is the results from the complex interplay between the protein structure embedded in the membrane called ion pumps or ion channels. Okay, this ion pumps are allowing some ions to pass through them. That means the sodium can pass inside, potassium can go outside. Because of the movement of these ions, the types of ion channels in the membrane usually vary across different parts of the cells, okay? Giving the dendroids, axon, and cell body different electrical property, okay? That means in the neurons, what are the parts of the neurons? Neurons have some parts too. In last class we studied important parts.
Cell body axon.
Right. Okay, cell body axons. Okay, all these parts have different types of ion channels. Okay, that's why they are having different electrical properties. Okay. Because of this, some part of the membrane are excitable. They can generate the action potentials, whereas others are not. The most excitable part of the neuron is usually the axon hillock. So axon hillock is the point where the axon leaves the body. So this is the most excitable part of the neuron. But the axon and cell body are also excitable in most cases. The highest excitable part is axon hillock. Each excitable patch of membrane has two important membrane potential. There are two important membrane potential at these excitable regions that are resting potentials, which is the value, the membrane potential as long as nothing perturbs the cells, and the highest value is called as threshold potentials. Okay, this simply means the excitable patch of membranes has two important level of membrane potentials. First one is the resting potential. Resting potential means the voltage of the neurons when there is no impulse pass through the neurons, that is about minus 70 millivolts. And these parts are also having threshold potential, that is minus 55 millivolts. Here, the synaptic input to a neuron. Okay, the synaptic inputs to a neuron causes the membrane to depolarize or hyperpolarize, and this causes the membrane potential to rise and fall. Okay, this is all happening when the channels of sodium and potassium is opening. So the reason is synaptic inputs. The synaptic inputs are causing the membranes to depolarize or hyperpolarize. Okay. Action potentials are triggered when there is sorry. Action potential are triggered when enough depolarization accumulates to bring the membrane potential up to threshold. That means initially there are minus 70 millivolts, and when the sodium ions are coming inside, they are reaching the threshold levels minus 55 millivolts, and then the action potential is triggered. So when the action potential is triggered, the membrane potentials actually shoots upwards. That means you can see on the graph that is depolarization occurs, and that will reach plus 100 millivolts, and then equally shoot back downwards, that is repolarization, and often ending below the resting levels where it remains for some period. Okay, I'll show you the pictures. So this is the picture. So here, this is the minus 70 millivolt, that is stimulus. And this is the threshold level, that is minus 55 millivolt. And this is the depolarization because of the intake of sodiums. And this is repolarization, this is hyperpolarization. Okay. I already explained this graph on the last class. And again, the same picture is, the same concepts is repeating. Okay, this is the last slide. So before considering the propagation of action potentials, propagations means the transfer or movement of the action potential along the axons and their terminations at the synaptic knobs, it is helpful to consider the methods by which the action potential can be initiated at the axon hillock. So here, now we are going to see about the initiation of how the action potential is starting at the axon hillock. The first requirement is that the membrane voltage at the hillock, axon hillock, be raised above the threshold for firing. Okay? So the first important requirement is the membrane voltage should pass or should exceed the threshold values. There are several ways in which depolarization can occur. So please note this picture. We are going to study about these pictures, and this is the axon terminal, and this is synaptic cleft, and this is dendritic spines. So when an action potential arrives at the end of the presynaptic axon, okay. So this is the presynaptic axon when action potentials are coming, reaching the presynaptic axon. This is in the yellow color. We will study what will happen. It causes the release of neurotransmitters, so molecules that open the ion channels in the postsynaptic neurons. Okay, action potential, when the action potential is arriving the end of the presynaptic. Okay, please don't confuse with pre and post. Presynaptic axon, it causes the release of some neurotransmitters. Okay, that is opening the ion channels in the postsynaptic membranes. So this is the presynaptic membranes. The action potential causes the release of these neurotransmitter receptors, and these are the neurotransmitter receptors, and these neurotransmitter receptors are opening the ion channels at postsynaptic membrane. So the postsynaptic neurons. So the combined excitatory and inhibitory postsynaptic potential of such inputs can begin the new action potential in the postsynaptic neurons. So here after ion channel is opening on the postsynaptic neurons, the membrane potential, so action potential begins there. So this is the picture. This is a presynaptic membrane and this is a postsynaptic membrane. And when the action potential reaches here, the voltage channels of calcium will open, and this calcium causes the release of these neurotransmitters, and that will cause the voltage chemical gated ion channels to open. So here the process is explained at the end of the presynaptic neurons, where the voltage gated calcium channels, when an action potential reaches the synapse, this channel open and causing the calcium ion to flow into the cells. This calcium ions causes the synaptic vesicles to fuse with the cell membrane, releasing the contents, the neurotransmitters chemicals. The neurotransmitters will diffuse across the synaptic cleft, and then these neurotransmitters bind to the neuroreceptors at the Can you all see the slides?
Yes, ma'am.
Thank you so much. So the neurotransmitters which is released from the presynaptic membranes will bind to the neuroreceptors. Receptors means that is receiving those transmitters. These receptors are available on the postsynaptic membrane, and this is causing the channels to open. In example, shows the sodium channels can open, so sodium ions flow in. Okay, this causes a depolarization of the postsynaptic membrane, which may initiate an action potential and the threshold is reached. If the threshold is reached, the neurotransmitters is broken down by the specific enzymes in the synaptic cleft. Okay, I'll explain it again. So it's a simple concept. When the action potential reaches here, the presynaptic membranes, these channels, voltage gated calcium channels will open and the calcium ions will flow inside. Okay, this calcium ions will be responsible for moving the synaptic vesicles. This is the synaptic vesicles that are containing neurotransmitters. Okay, here this synaptic vesicles will come join with these membranes and it will release the neurotransmitters outside. Okay, these neurotransmitters this will cause the opening of ion channels in the postsynaptic membrane. So when the sodium ion channel is increasing, the sodium ions can come inside. If the threshold level is reached on the postsynaptic neuron, then the action potential may begin. And these neurotransmitters will be then broken down by specific enzymes in the synaptic cleft. For example, acetylcholine esterase breakdowns the neurotransmitter acetylcholine. The breakdown products are absorbed by the presynaptic membranes again by endocytosis and used to resynthesize more neurotransmitters using the energy from mitochondria. This stops the synapse being permanently on. Okay. Now we have completed the subject action potentials. Anyway, on the physical class, maximum I will try to explain everything again. And there are just okay, if I'm having time, if the physical class is not possible, then with whiteboard I will explain. Okay, I can take this whiteboard in the Zoom, and with whiteboard I'll try to explain it again anyway. It is endocrinology. Okay, this is other practicals. So please note this picture. Okay, you can take screenshot or you can get better pictures. So if it is not clear, you can take the picture from online. I think it's somewhat clear. So in this picture, you can see some of the important parts of the brain: cerebral cortex and pineal gland, hypothalamus. This is the most important part regarding endocrinology. The hypothalamus, pituitary, that means the pituitary glands. There are two types: anterior pituitary glands and posterior pituitary glands. And other part is optic chiasm, which we'll study later, and suprachiasmatic nucleus, the other region. Okay. So please have a look at this picture sometimes. This is the hypothalamus. Okay. The hypothalamus, it is connected with the pituitary glands. Okay. So this one is the anterior pituitary glands, and the behind one is the posterior pituitary glands. Okay. And these are just some other portions: optic chiasm, infundibulums, median eminence. Okay, we will study about all these parts detailing coming classes. So these are some relevant pictures, and here we can start the class. Pituitary glands and its relation to the hypothalamus. Okay, the pituitary gland is always related with hypothalamus. The first point, okay, if you want, first you can note down all these things, so you can please read this slide for about 1 minute, and then I will explain. Okay, about this gland, it is also called as hypophysis. Okay, another name. And about the dimensions, it is about 1 centimeter in diameter and having 0.1 to 1 gram in weight. It is located at the base of the brain. Okay, the fifth point is much important point. The pituitary gland is connected with hypothalamus. Okay, by pituitary stalk. This is important. Okay, the pituitary gland is connected with hypothalamus by pituitary stalk. It can be divided into two distinct portions: anterior pituitary region, posterior pituitary regions, and between this anterior and posterior, anterior means front, posterior means backwards. Okay, between this anterior and posterior, they're having a relatively avascular zone called as pars intermedia. Okay, this avascular zone is absent in the human being, and but it is much larger and much more functional in some lower animals. So here, in this picture, you can see this is anterior pituitary and this is posterior pituitary glands, and this is the pituitary stalk. Okay, the pituitary gland is connected with the hypothalamus through the pituitary stalk. So in this picture is also, again, you can see this is hypothalamus and this is posterior pituitary gland and anterior pituitary gland. So again, okay, I'll ask some questions in the slide. Okay, which part is connecting the pituitary glands with hypothalamus?
Pituitary stalk.
Okay, very good. Okay, This is hypothalamic, anterior pituitary, and target organ hormones. The very important chart, okay? These are the anterior pituitary hormones that is secreted in anterior pituitary gland, okay? You may already familiar with these hormones, growth hormone, GH, TSH, thyroid stimulating hormone, ACTH, FSH, LH, PRH. Okay, and hypothalamic hormones are growth hormone, releasing hormone. This GHRH, growth hormone. Okay, and TRH is secreted by hypothalamus. CRH, GnRH, these all are secreted by hypothalamus. And on the other charts, you can see the target organs, and on the final column, you can see primary target organ hormone or mediator. Okay, these first three columns are very important. You must know anterior pituitary hormones, and you must know the respective hypothalamic hormones, and you must know about the target organs. Where it is target organs means where these hormone is functioning. Okay, pituitary hormones. There are 6 hormones on the anterior pituitary that plays a major role in the central metabolic functions throughout the body, such as growth hormones, adrenocorticotropin, TSH, prolactin, and gonadotropic hormones. So, under the gonadotropic hormones, the FSH, follicle stimulating hormone, and LH is coming. The 2 hormones that are secreted by the posterior pituitary player are the roles. Okay, here these hormones are just secreted by the posterior pituitary gland but produced by hypothalamus. Okay, the hormones of posterior pituitary gland is produced by hypothalamus and secreted by the posterior pituitary glands, such as ADH or vasopressin and oxytocin. Okay, please go read the slides for about 2 minutes. I'll explain later. Okay, these pituitary hormones. First one is GH, growth hormones. It's obviously promote the growth of the entire body, which you can understand by its name. And it is affecting, the growth is promoted by affecting the protein formation, cell multiplications, and cell differentiations. Okay, simply the function of the growth hormone is to promote the growth of entire body by affecting protein formation, cell multiplication, and cell differentiation. ACTH that controls the secretion of some of adrenocortical hormones, okay, which affect the metabolism of glucose, proteins, and fats. And find TSH, and that is control the rate of secretion of thyroxine and triiodothyronine by the thyroid glands. Okay, and these hormones are control the rates of most intercellular chemical reactions in the body. Okay, growth hormone simply it is promoting the growth. Okay, it's promoting the growth by affecting protein formation, cell multiplication, cell differentiation. ACTH, it secretes the adrenocortical hormones that can affect the metabolism of certain nutrition such as glucose, proteins, and fats. Okay, ACTH that is controlling the secretion of some adrenocortical hormones. TSH control the secretion of thyroxine and triiodothyronine. Okay, these hormones control the rate of most of the intracellular chemical reactions in the body. You must know all these pituitary hormone names and their functions. Okay, now can anyone tell what is the function of growth hormones?
Promotes the growth of the entire body.
Okay. By which mechanisms they are promoting it can promote the growth?
Cell
How it
multiplication.
is promoting the growth, just like that. Protein formation, cell differentiation, cell multiplication. Okay. Prolactin. So this promote the mammary gland for the development and milk production. Okay. Prolactin. Okay, I think it is. Oh, we are studying about the prolactin, and it is promote the mammary gland and development, mammary gland development and milk production. So for both. So in mammals, for mammary gland is responsible, and FSH and LH. Okay, follicle stimulating hormones and luteinizing hormones. They control the growth of the reproductive organs such as ovaries and testes, as well as their hormone and reproductive activities. So all this, their growth, hormonal activities, and reproductive activities are controlled by FSH and LH. ADH, okay, that is controlling the rate of water excretion into the urine. Thus help to control the water concentration or in the body fluids. Simply for to maintain the osmotic balance, ADH is very important, and that is controlling the rate of water excretion into the urine. Okay, oxytocin, that is posterior pituitary hormone. ADH also posterior pituitary hormones, and here this helps to express the milk from the gland. That means help for the contraction of the alveoli and tube. Express milk from the gland of the breast and possibly help in the parturition of the fetus. Okay, it's also help to contract the uterus and assist in the parturition of the fetus. And this is some of the cells that are available on the anterior pituitary hormones. So this anterior pituitary gland, this anterior pituitary gland contain different cell type that synthesize and secrete hormones. Okay, there is one cell types for each major hormone. So we know G, main growth hormone. What is the main function of growth hormones? You just answered now.